one FIP online and another offline. And this will be in online mode. And I will be very happy to address you. It's an interactive session. And anything, any queries or anything, you please stop me and you can. Otherwise, even after the seminar also, you can. After this talk also, you can. Anything you feel free. And because my specialization is and with my limited knowledge, I'll see whether I could give some information. And even as Dr. Nanjan Swami sir told, so I just wanted to bridge university with higher education with the public, or university with the public, and that too in local language, Kannada. So thereby it will reach even layman. And that's a prime duty of all teachers. Basically, whether we are assistant professor, associate professor, director, professor, whatever, we are all teachers. So, and we have a prime duty, and our role in society is amicable. It is commendable, and we can change the march of our youth to productivity. So with this brief introduction today, I will just give the brief outline of infertility. You must be knowing nowadays infertility is a burning problem. And 25 years back, it was not a major issue. And we were thinking about population explosion in India and globally. And India and China, we were in the competition. And later on, family planning, all these things came. And all of us, we have 1 or 2 kids. And without any this one, no. So every family, they knew that the importance. But now, this is the this next another future world. So there is a problem. There is a threat for human survival only. Not the population explosion. There is a problem of infertility. And I always used to tell everyone that another 10 years, we have a threat of artificial intelligence, machine learning. Probably that will replace human resources, all robotic thing. Other than that, another important threat is a mental health. So we have to struggle a lot to have a mental health major issue. And right now we are not knowing, we are in a race for our goal, achievement, academic thing, all this. But in this thing, in this race, we are losing our health. And stress is one of the important thing which causes male infertility at present context. There are many other things, our diet, lifestyle, hobbies, all these things. Other than that, very important component is stress. Stress causes erectile dysfunction in men. And that is another important thing, one of the risk factor in male infertility. So today we unravel the roots of male infertility. What are all the reasons, how the person will become infertile, all these issues. So infertility, it is defined as a failure to achieve a pregnancy after 1 year of regular unprotected intercourse. That's a WHO definition. So with the same partner, that's also more important, without using any family planning protection. Even with the regular intercourse, if they are unable to have issues after 1 year, then that is called as a couple infertility. We'll not be knowing whether male has a problem or the female has the problem. Later on, when we do the diagnosis, then we'll come to know that whether there's a problem occurs in the male or in the female, or sometimes both will be having the problem. Females might be having a PCOD problem, males will be having an erectile problem, or many other things. So it might be in the either in the male or in the female, or in both of them, the couple infertility. But nowadays, if you look into the statistics and research articles, no, they say researchers authenticatedly, they claim that male infertility in male is more than the female infertility. 60% of the infertile couple, among them, male having the issues when compared to female. We'll see why male is a susceptible group now for infertility condition. And I will discuss how it will happen. So the incidence in the globally as well as in India, it is estimated that 60 to 80 million couples suffer from infertility every year. This is the every year data. It is around 60 to 80 million, and 15 to 20 million in India alone. We have this. And if you take the percentage, it is 0.74%, but it is increasing. Year by it is increasing, and it is not a small number also. So let me give a brief outline. What are the conditions in infertility? Generally, each individual have a different type of condition. And it is not confined to one person. If one person, one couple, if they come for the diagnosis, no, they might have oligospermia. The male has oligospermia, astenotereto, all these conditions. It varies from individual to individual. So the generalized condition for male infertility is oligospermia. You might heard about this terminology, oligospermia. Means there is a reduction in the sperm count. That is, if it is less than 20 million per ml, then it is called as oligospermia. 50 years back, there was a survey done globally. It is a multi-centric study, involved different countries. And they have analyzed, they have that record also. You can go to research articles now. Definitely you will get the record. So earlier, 50 years back, a normal human male has the semen count of 200 million per ml. It was a 200 million per ml 50 years back. Now they say normal is more than 20 million. From 200, one digit is removed. 200, it has reduced. It became 150, then 100, then 50, like that. Now, I have analyzed more than 10,000 couple. My entire career, 14, 15 PhDs are on infertility only, related to that, at molecular, from the grassroots level, from the scratch till molecular level. All the level we have analyzed the reason for the male infertility. Almost 10,000 couple I have analyzed, and I got to know both the normal as well as infertile. Even in the normal, hardly we can see healthy male having 80 million or 60 million, 50 million, not more than 100. 100 or hardly you can get one or two people more than 100 million per ml. Otherwise, there is a compromise in the sperm count in males everywhere, not only India, everywhere. There are a lot of reasons. It is most of the time it is an environmental factor. So oligospermia means there is a count is reduced to less than 20 million per ml. Azoospermia means spermatozoa were absent. The semen is a fluid which should have the spermatozoa. But in an azoospermic condition, there is no spermatozoa at all. Spermatogenesis could happen. Sperms were produced, but they are unable to mature in the epididymis. You must be knowing in a male, primary reproductive organ, secondary reproductive organ, or accessory organ. Primary is a testis, where the spermatogenesis happen. Secondary are accessory, testis and epididymis. Secondary accessory glands are like a prostate, seminal vesicle, vas deferens, all these things. So here there is a spermatogenesis. In some of them, there is a spermatogenesis, but still it is unable to mature. Or some of them, they don't have the vas deferens. Vas deferens is a connection, which it is like a passage for the spermatozoa. But what happened? There is one condition in genetic condition. It is called as a CFTR mutation, cystic fibrosis. Cystic fibrosis is a condition where the person doesn't have the vas deferens by birth. So what happened, though there is a spermatogenesis, then sperms, they won't be there in the semen. Like that, in some individual, there is no spermatogenesis at all. There are a lot of such variations are there. Ultimately, azoospermia, spermatozoa were absent. Sometimes there is a retrograde ejaculation. In retrograde ejaculation, sperms will go through urine. It won't come into semen. It will pass on outside through urine. So that is because of the blockage. So there must be some blockage in the passage. Thereby, it is a retrograde. That is called as a retrograde ejaculation. So next condition is the asthenospermia. There is no motility. Always, you can, for most of these videos related to these things, you can go to Math with Malini. As sir mentioned, there is a YouTube channel that in Kannada, and more than 380, 368 videos are there related to all the aspect. And even in one of the YouTube channel, Gauri Shekki also you can view there. All the some of the information I have given. So thereby you can, I need not explain all these things again and again. No motility. Always I used to say, human body is a mesmerizing thing. It is a greatest creation because all the pathways, all the how these genes will be expressed, everything is a mesmerizing and amazing. And whenever I read all molecular level pathways and these things, no, I wonder every time it is it is a wonder for me to analyze the things. So because if there is no motility, if you have seen the spermatozoa through microscope, you really wonderstruck. So such a fast, vigorous movement the spermatozoa has, you can't imagine. Is this the beauty of the life? You never, because we have seen the spermatozoa head and tail, all these things, but how they move, it is unimaginable thing. So by chance, if they lose the motility, then it is called as the asthenospermia condition. Then there's another condition called as a teratospermia, means abnormal morphology. The shape of the spermatozoa is completely abnormal. So generally it will be have head and tail. Sometimes the head will be different. Tail, double tail will be there. Tail is short. So any of these changes, no morphology, then it is unable to penetrate the zona pellucida and fertilize and do the fertilization. So teratospermia is also another condition. Oligoteratospermia means low count with abnormal morphology. Oligoasthenospermia means low count with low motility. Oligoasthenoteratospermia means low count, abnormal morphology with a low motility. This OAT condition is horrible. And no treatment, even there is no improvement in the treatment. So they go for a IVF. They can go for a donor sperm. And this oligo borderline, if it is a borderline, that's why I used to say, youth, before you get married, no, you better go for a pre-marital medical checkup. Just get your sperm count done. If there is a reduction or low count, then before you marry, you just take treatment and thereby you can improve your sperm count. Otherwise, what happen? No? After the marriage, no issues. Then they'll come to know that boy has a problem. And if it is a rectifiable, fine. If not, it is always, nowadays the family has become very sensitive. We don't know how, when it will break. All these things we have seen among this generation. They are very sensitive. They don't tolerate anything. Infertility condition, both in male or female, nobody will tolerate. So all these things, it could be done. It could be diagnosed. That's why premarital medical checkup is very, very essential for the sperm analysis, or even hematological studies, like they can check for a HIV, HTLV, or even they can go for a sexually transmitted disease, syphilis, gonorrhea, syphilis, chlamydia, all these infections, no, they can check and get the treatment. If there is any problem, generally it won't happen, but if there is anything wrong, then they can get, could be rectified. So these are all the different conditions. It's not moving. Excuse me 1 minute. ಇದು ಮೂವತ್ತಿಲ್ಲ ಮೂವತ್ತಿಲ್ಲ ಸ್ಲೈಡ್ಸ್
ಅದು ಸ್ವಲ್ಪ ಜಾಸ್ತಿ ಗೊತ್ತಿದಾಗ
ಓಕೆ ಯಾ ಎಂಟ್ರಿ ಪ್ರೆಸ್ ಮಾಡ್ಕೊಂಡು ಮಾಡ್ಬೇಕಾ
ಮ್ಯಾಮ್ ಏನಿಲ್ಲ ಇದರ ಮೇಲೆ ಒಂದ್ಸಲ
ಕ್ಲಿಕ್ ಮಾಡಿ ಓಕೆ ಯಾ
ಅಷ್ಟೇ
okay so we'll see what are all the etiologies means a causative agent. One is anatomical, maybe some change in the testes, epididymis, vas deferens, seminal vesicle, prostate, that's anatomical. Some problem in the structure of the reproductive organ. And sometimes what happen, there is a tumor in the reproductive organ. There is because of more infection, there might be some calcification. Always we neglect urinary tract infection, and neglected urinary tract infection make some clouding in the all this reproductive organ, thereby it will be calcified. So and it start degenerating the cells, both in the testes or in the prostate, wherever. So secretion will not be there, sperm production will not be there. So anatomical tumor, infection, or by birth they have some abnormality, all these things. Genetical means there may be mutation in some of the genes which are involved in the spermatogenesis, or maybe changes in the chromosome. My entire career, entire academic thing, research is mainly on this chromosome molecular related aspect. Then even I used to tell everyone that I have screened a lot many people and interacted with the transgender female, transgender male, and lesbians, gays, all these hermaphrodite, pseudo hermaphrodite, all kind of people, and even Klinefelters, Turners. There are lot of such thing. Within fraction of second, when I interact with anybody, I can some extent guess that there is something wrong with the individual. So it might be my guess. Later on, for the confirmation, we have to go for some analysis. So definitely, and nowhere I have gone wrong all these days. I have interacted with many people. One best example, once you must be knowing Vasudendra. He is a Kannada writer. He is a fantastic writer in Kannada books. So he had visited our centers once, a physical mode. And once he has entered, immediately I could sense that something wrong in his gesture, a kind of thing. Immediately it came to my mind, but I didn't reveal, and all these things are very confidential in human genetics. So we took them to the this one participants, and he has introduced himself, and he mentioned that he is a gay. So the kind of thing. And then I thought I have not gone wrong. My scientific analysis has not gone wrong, because within no time, just he has entered, how he has entered, I got to know that he has a problem. And it was confirmed. Means genetic problem, lot of such things will be there. There is a change in the chromosome number, and a lot of mutations in the genes which are involved in spermatogenesis, sex orientation genes, all these epigenetic phenomenon, so many things will be there. Next is the endocrinological. And you must be knowing hormone plays a very important role. Either it's a pituitary hormone, hypothalamic hormone, pituitary, then pituitary, you must be knowing it will release a TSH, FSH, LH, all these things. And because of this, testosterone will be released by the testes, and even adrenal gland will release androgens. All these things, if there is anything hyper secretion of these hormones, follicular stimulating hormone, luteinizing hormone, TSH also important, and testosterone is important. And sometimes the inactive form of the testosterone has to be converted into active form of testosterone. Dihydrotestosterone has to be converted into testosterone, and the enzyme which is involved in this pathway, if it is absent also, the pro condition will be there, but active form will not be there. So finally, whatever may be, if there is no sufficient testosterone, then the person will have the problem of libido, erectile dysfunction, even spermatogenesis, everything he will face. Then I told you immunological problem. There is a production of anti-sperm antibodies sometimes in our body. You must be knowing autoimmune disorder like rheumatoid arthritis, psoriasis, then alopecia, so many these things. No, nowadays even have given very good information about why earlier this rheumatoid arthritis was 1 in 1 lakh, very rare. But nowadays all are telling I have autoimmune problem, and they are taking steroids. And what's wrong with the people? When we start analyzing, there are so many other factors involved. So why they are developing all this thing? We are losing health, that is for sure, because of so many other reasons. Then gradually we'll come to know that there are a lot of other issues. And this immunological problem is body itself produce antibodies against the spermatozoa. Against the sperm, body itself will produce antibodies, and antibodies will kill the sperms. So thereby those individuals, they don't have. And it is because of the breakage of the blood testis barrier. We have different barriers in our body. That's the blood brain barrier is there, blood testis barrier is there. So if there is a breakage in the blood testis barrier, then that's why I used to tell everyone, be patient, go, be very slow and study. Nowadays, no youth, they don't listen. Within 1 or 2 months, they need the 6 pack, 10 packs, all the packs. But what happened? Anything, it should be very slow process. So even you can have pack, but you take 2 years, 3 years, nothing wrong. But within short span, no, what happened? Because of the over this thing, exercise and strain, 8 hours, 10 hours in gym, there's a torsion. There's a breakage of the blood testis barrier. Then what happened? All the antibodies, the sperm will be entered into the blood, and body will think it is a foreign material and start producing antibody. All these things will happen. Sometimes accidents, and lifting a heavy thing, that is people, weightlifters. There are so many things even that causes a immunological problem. Sometimes the mutation also. Sexually transmitted diseases, I told you, syphilis, gonorrhea, chlamydia, HIV, HTLV, hepatitis, all these things, fungal infection, yeast infection, so many are there. Even that will cause the infertility. Parasitic, but these are all the treatable thing. Parasitic disease, even malaria also causes infertility. Malaria, there are a lot of reports saying that malaria causes infertility. There are many such vector-borne diseases. The trypanosoma, which causes sleeping sickness, even that causes infertility. It compromises with the fertility factor. Exposure to potentially toxic substance. This is now, we are in the world of chemicals. Everything, coloring agent, preservatives, food additives, you name anything, it has a chemical. It has either insecticide, pesticide, herbicide, fungicide, all these preservatives, all these have some impact on our body. And I used to tell everyone, don't take even paracetamol, just like a peppermint. Dolo 650, no, for everything we take. We don't always care for our body, and always, no, it is a gift by God, such an amazing, wonderful thing. And small thing, headache, fever, no, immediately they take a tablet. So body has its own healing power, but give some time, relax, sleep well, eat well, body will heal by itself. And without animals, no, there is no need of such a treatment. They know how to heal their own body if there is something wrong. But nowadays, no, without thinking, simply we consume all paracetamol, painkiller, everything, just like a chocolate peppermint. And it is like a hobby. Some people know small headache, you just leave it for some time. How much you can tolerate until then? There is a nausea, you will vomit, fine. Then you will be relaxed. But why you suppress the healing process in the body? Exposure to potentially toxic substance, please avoid plastic. Plastic will release dioxin, phthalate, very potent xenoestrogen. Future world, now already you have seen more of homosexuality in our society. Earlier it was less. Nowadays, more of homosexuality, and they are coming up openly, and they are telling, I am a gay, I am a lesbian, all these kind of things we have seen. Why? Because there is all these chemicals, they are xenoestrogen. They will alter our endocrine pathway. Xenoestrogen, they act as an estrogen. Thereby, the males will transform gradually. No, physically they are male, but their behavior, their thinking, all these things, they started like, like a female. Once they think that no, female, then what happened? They always know they become a homosexual. Like that, there are a lot of such things will happen. Even during pregnancy, also there shouldn't be overload of testosterone or less of the testosterone on the fetus that will make the sex orientation. So idiopathic condition, these are all, I will give some more example. The etiology means causative agent. Idiopathic condition means everything normal, but still there is an infertile condition. Where at molecular level, we have to check which are all the gene. At gross level, all reports are normal. A hormonal report, a cytogenetic report, clinical examination, everything fine, fantastic on both of them, but still no issues. Means idiopathic. Idiopathic means the reason, still we have to unravel what is the reason for that. At molecular level, something wrong, the gene expression, or in the protein level, protein folding, or truncated protein, anything could happen. Okay. So this diagram, yes.
Ma'am,
Yeah.
is there any study between COVID and infertility? The COVID patient showing any form of infertility?
COVID patient, no, the study has not been done because for this we need to take the youngsters.
Okay.
Already issue means nobody will come forward. They have issues, no. So there is infertility is different from impotency. Infertility, they can't have the baby or the issue. Impotency means they are not involved in the sexual process. Erectile dysfunction, all those things are impotency. But infertile males are sexually active. Okay. So that's why somebody has to take, and we need to take a youngsters for this study to check whether. But what happened? No, pre-COVID, we have not done the sperm analysis, right?
Yes, ma'am.
Before COVID only we have to analyze. Then after COVID, what happened? We can. Now we don't know whether it is a true condition or effect of the COVID. So before, after, always any such studies, no. before the development of the disease, after the development of the disease, that will give the correct picture, and that will be compared with the control also. Control also before the COVID, and means they're not affected with COVID. Before the COVID, after the COVID, it might be due to the exposure also, the duration of the exposure or the dosage of the chemical, everything. So we need to plan such studies, but it is a sudden this one, no, so nobody has thought in that direction.
Madam, I agree. Along
Yes.
with IPSITA, so I think as you mentioned, even malaria
Yes.
has impact.
Yes.
So I think it is more apt. I think there may be effect of viral infection on fertility and also.
Many pathogens, you must be knowing, dental health has an impact on the infertility. Dental health.
Yes.
There is a condition called as a periodontitis. Periodontitis is a bacterial infection. The bacteria will travel from mouth, then it goes to heart, it causes myocardial infarction. It could travel down, go to testis, where it causes infection. If it is not treated, you can treat with a broad spectrum antibiotic, not an issue. But if it is not treated, it causes a permanent degeneration of the Sertoli cell or the spermatogonial cell. Then naturally the person will be compromised. But they never knew that the dental issue is the reason for the infertility. Nobody knows. We are most neglected about the dental health. But dental health is also very important for both for pregnant female as well. It has impact on developing fetus, dental health. So before they become pregnant now, they have to scan their entire body, everything normal, mentally, physically, socially, spiritually, all kind. Then only they could give birth to healthy baby. Here,
Thank you.
yeah, the reason impairment in normal spermatogenesis, I told you, testicular cancer, I told you, systemic disease, all metabolic disorder, even diabetic obesity, thyroid, high cholesterol level, cancer, all these systemic diseases has an impact on infertility. Because in a diabetic person, there is a damage in the nerve end. And the testis, it is a muscular. And because of the neuronal stimulation only, there will be erection. What happened in the diabetic, the nerve end will be damaged, thereby there is no erection in the severe diabetic males. Thereby they have to manage the blood glucose level always. Otherwise they have the problem of the erection. So that's a systemic disease. Then no erection means naturally it is an infertility also. Then hormonal dysfunction, I told you, and sexually transmitted testicular trauma due to torsion, also sometimes accident, and there are injury in the spinal cord also that causes a testicular trauma. Ductal obstruction, drugs, many medication, and for some diseases they need to take a medication for many years. Epilepsy, tuberculosis, there are so many other things, and even blood thinners also many people are taking. All these, any chemical inside, but for the survival we need that, otherwise we can't manage. But all these, any drug from outside has an impact. It will compromise. So we should check whether I should leave or die, or whether I should leave with some condition. That compromise we have to make a demarcation. And then genetical cryptorchidism means in some people know what happened. The testis has not descended down. The testis has to descend, and it will be in the scrotal sac. Testis will kept always outside the body because it requires less temperature than the body temperature. Body temperature is 37 degree, and the spermatogenesis happens at less than it is 34 36, 37, 34 or 33. At that condition, spermatogenesis it will happen. So in some males, no, by birth only. So if testes are not descended into the scrotal sac, then it is an infertility condition. And temperature is one of the reasons for infertility nowadays, because nowadays youth, they are using always jeans and tight innerwears, no aeration. And because of that, those who are living in a high temperature area. Earlier, our ancestors, they used to have in the high temperature area cotton thing, more aeration, pancha, dhoti, all those things, good aeration, good thing for the fertility. Now, all those cotton things are replaced by jeans. because of globalization. And so there is a compromise in the fertility also. And spinal cord injury, I told you, varicose vein, especially teachers, those who are standing for long duration, they develop the varicose. Okay, varicose vein in the testis also, definitely, because it's an extra vein and more temperature will be there. So it compromises the spermatogenesis. Environmental factor, I told you, and retrograde also ejaculation. So why this study is very important? Because I'm the first person, I made this attempt and long back, and I started taking the samples, because asking blood with anyone, blood sample for the analysis is fine. But semen sample, if I ask anyone, they will run away. So nobody will cooperate with me. So I tied up with some IVF centers, and for control, I need a healthy individual. So it is a really Hercules job, very tough job. I could convince them, and different modalities, ways and means by which finally they convince, and they give the sample. Even though ample of infertility cases are reported, but so far no systematic studies have been done on the genetical, biochemical, endocrinological, and overall picture, immunological basis of male infertility. And many few research centers like CCMB, CDFT, even you must be knowing Dr. Tangaraj from CCMB and CDFT. Also, there are few scientists. They were in IIC. They work on a mouse model, but on human, only 1 or 2 people throughout the country they are working, but not on the complete picture, either on the biochemical pathway or the endocrinological or the genetical or something like that. But with that bit and pieces, we don't get the exact picture. If you want to know the exact story, how it has started, how it happens, how it is progressing, we need to have the knowledge from all round corner. It is a holistic approach, not a single approach. Single approach won't give you any message. When you start looking the things, problem holistically from all angles, then definitely we come with some solution. We could develop some markers, we could develop some diagnostic kits, such things will be done. So present such human studies on human infertility, which help us to establish the etiology. We could make out what are all the causative agent. So in our population, in Indian context or in Karnataka, what are all the reason for male infertility? Because it may not be similar in a Western population. Indian data is completely different from Western data, because we have a lot of inbreeding, we have many caste, community, subcaste, region, religion, practices, so many variation. Thereby, and more of inbreeding means marriage between the same community and relatives. Because of that, some of the diseases will be transmitted from generation to generation. So like that. That's why the etiology of male infertility in Indian context, that in South India, is completely different from the global statistics. So we can understand the prevalence, how prevalent it is, type of anomalies, which are all the anomalies are common in our population. Thereby, we could region-wise, we could make out. For example, the Kasaragod belt region, there is a endosulfan, you must be knowing, endosulfan tragedy. And even the endosulfan will affect the fertility of an individual. That region more of abortion, in the sense recurrent pregnancy losses and newborn death will be there, and anomalies in the baby and infertility. So region wise, religion wise also we get some idea why such things are happening. Type of anomalies, degree of relatedness, and based on all these things, finally, just analyzing the problem is not the end solution. We should give some solution to the society. Then only the story will be completed. Otherwise, I did this, I did that. It's not the final solution. I should give some message. Take home message for everyone. Oh, this is the message from this study. Like that, I should give. Then only it is in a applied aspect. That's the applied one. Whatever research in life science we are doing, finally it should have some application either on animals or in our society or somewhere. Someone has to be benefited from this. So in order to do that, it is a Herculean job, I told you. And I am giving the outline. So each factors will be for some thesis work, but I'm giving the outline how one could analyze and how we have analyzed. So first of all, we make an establishment of genetic registry. We collect the information. Every information is important for me. I shouldn't neglect that this is not related to me. This is not important. At that point, it may not be relevant, but in future, the data might be relevant. At that point, I may not be having some concept or hypothesis, but if you have the data, then definitely after some years, if you read some research articles, you could connect that problem with the existing. So, but that time you may not get the same individual, same person for the analysis. So you have to collect maximum information about the person. Pedigree will be done. Pedigree means it is a systematic representation of the generation, the 3-generation pedigree, 4-generation pedigree. So thereby we can make out whether such things are there in the family. And then what is the inheritance pattern, all those things. Then chromosomal analysis. You must be knowing female XX, male XY, and Y chromosome is important because of the SRY gene, all those. So chromosomal analysis we have to do. Then analysis of the sperm sex chromosome aneuploidy. Then there is you must be knowing there is a aberration, chromosomal, numerical, structural, all these things result into problem. Analysis of the semen quality, very important, gold standard technique. This we have to do, even though you are working on molecular aspect, some of the important baseline work we shouldn't neglect. So sperm function test, then evaluation of oxidative stress parameter in semen, biochemical analysis of the semen plasma, immunological analysis, hormonal analysis, molecular analysis, ultrasound scanning. So entire thing we need to do. Thereby, we will be knowing what exactly the problem and the extent of the problem and even solution for that particular problem. So in here, I am just taking a 250 because I can't include 10,000 samples here. 250 control, 250 healthy, 250 infertile males will be selected. And human genetic study, always we need to take a consent letter from the patients. Otherwise, we will be sued. Somebody, our enemies, no, all our people, they do backstab once we are through with all these things. So it is there is a ethical committee will be there, university ethical committee, hospital ethical committee, and we need to present our proposal, work there, and many people will be there. One advocate, one social science person, NGO, then scientist, many will be there. They scrutinize and they check how we are handling the sample, all these things. So that will be cleared and consent will be taken. And I need to tell each one that this is your data will be used for research or it is for the publication. And if they have a consent, then only we can collect the blood sample, semen sample for the analysis. And genetic registry, I told you, what are all the information we will collect from the genetic registry? We will collect the family history. We take the medical history, reproductive history, and lifestyle. And you must be knowing the human mentality. One round of interaction, nobody will reveal anything. We have to have a develop a confidence. Then only they will reveal what are all the things happened. It is just like a counseling them. So one round of counseling, they don't open up. We need that comfortable thing, confidence, then only, and they should be assured that the data, the identity will not be revealed, data will not be revealed, all these things, then they give the information. Otherwise, 30% of the information will be false. If I ask them, are you alcoholic in one sitting? No, they don't reveal. Are you a smoker? Are you, things like that. Or if you had some sexually transmitted disease, then nobody will open up. So we need to interact 3 to 4 sitting times, and interaction will be carried out with the infertile individual and with the family members to collect the authenticated information. Otherwise, the information might be wrong. So in human thing, it is not like animal study because it is based on the questionnaire. The information collecting, the 30% error will be there always. Only 70% we can guarantee. Pedigree analysis, I told you, it is a diagrammatic representation of the family relationship. and it is a visual tool for documenting the inherited disease. I told you, cystic fibrosis, it is a genetic disorder. There are a lot of such genetic disorder that is associated with infertility. Genetic disorders associated with infertility in female, PCOD. It is associated, they can't have the baby because anovulatory condition, no menstruation, no egg production, nothing like that. More of androgen will be there, all those things. So it is a documentation to check the inheritance of the disease. And lot of studies done, 10 to 15% of severe male infertility include chromosomal aberration, single gene mutation. All these things are associated. Lot of studies with the sperm defect with a diverse inheritance pattern also. So this is the pedigree. This how first generation, second generation, third. First generation, second generation, third generation, fourth generation. Here there is a infertility. And this male is an infertile male. Thereby there is no issue in this fourth generation. So similarly here, no issue here, no issue, no issue, like that. So here in another one, no issue, rest of them. So we have to put this pedigree and we have to cross check. And we can see 7% of the infertile male family history of infertility and show positive association with the consanguinity and male infertility. Consanguinity means it is a marriage between the relatives. Always, as a genetist, I never entertain consanguineous marriage or marriage between the relatives. Whether it's a first cousin marriage, uncle niece marriage, or whatever may be, no consanguineous marriage. If you want to have a healthy baby and baby with a good IQ, good EQ, good AQ, all these Qs, then you should go for outbreeding, not in the family, because we don't know how many genetic disorders we harbor in one copy. If both the alleles are defective, then it will be expressed as a disease. But one allele, one gene, one allele, then another partner will be normal. The lady look like normal, the male look like normal. But once they marry among the relatives, the baby will be getting, both of them are heterozygotes. Baby will become homozygote for the disease. And the disease will be, most of the time it is autosomal recessive. Recessive means both the copies are required. Autosomal dominant means just one copy is enough for the expression of the disease. So no consanguineous marriage at all. No, because that in the worst condition, they have to go for a that's carrier screening. So then once they are through with the carrier screening, find everything normal, sperm count, everything normal related to then or even other genetic disorder, then they can go for in a inevitable condition. Okay, we'll see how the semen analysis will be done. First of all, semen analysis is one of the important diagnostic tool to assess the fertility status of the male. Generally, what are all the things done in the semen analysis? We do the physical examination. That's why we should be self-examiner for our body. That's what they say, even in a cancer. Breast cancer, female, they have to go for a self-examination, whether there is a tumor or something in the body. Then self-examination is the best examination. Always, we should check what is the color of the sputum. What's the color of the urine? What's the color of the bowel? What's the color of the male's semen? The color itself indicating that something goes wrong. The physical examination, I will tell you what are all the indication. Coagulation means it will get gel. Liquefaction means the gel will become liquefied. pH should be neutral, 7. Color should be, I will tell you, it is like a grayish, white grayish color. Viscosity means it shouldn't be too much viscous. Viscous means that's it is like a sticky. So it shouldn't be. Then the volume should be more. So these are all physical examination. Just by looking into the semen after ejaculation, we will look into that and definitely it will tell you what is wrong with the individual. You need not go for molecular, you need not go for all these things. You could suspect at the grassroots level what could be the reason. Then you can deviate or you can channelize to look into what are all the problem happen in that particular individual. So coagulation means Once semen is ejaculated in a liquid state, it will gel shortly. Semen is a liquid form once it ejaculates, but immediately after exposure to air, it will become gel. If that is called as a coagulation. If there is an absence of coagulation, it is an indication of congenital absence of vas deferens and seminal vesicle. The person may not be having vas deferens or seminal vesicle. It is a congenital by birth. So that's the indication of the coagulation. Liquefaction means this gelled semen will be liquefied after 30 minutes. If you leave that one after 30 minutes, first it will be liquid. Then exposure, it will become gel. After 30 minutes, the gel will become liquid again. At room temperature, then it is called as a liquefaction time. And if there is a problem in the liquefaction time or the liquefaction, it indicates the dysfunction of accessory glands such as a prostate. Something wrong with the prostate. Nowadays, female breast cancer is a dominating leading cause of death in females. Males, leading cause of death is a prostate gland cancer. Majority prostate enlargement after 60s, everyone, earlier it was less, it was there less, but now there is a prostate enlargement and tumors in the prostate, they remove the prostate. Once prostate is removed, no control for urination, and you must compromise with many of our things, excretory activities. Odor, normal sample smell, which some find it unpleasant. Occasionally, a sample may smell spice such as a garlic and clove, which is quite normal. A kind of a pungent, not very pungent. It is not unpleasant, it is a pungent, it is a normal. A malodorous or unpleasant pungent smell is an indicative of infection. If it is intolerable, some odd smell, then it is infection. While an odorless semen is associated with abnormal prostate. No smell at all, it's a prostate. There is a prostate problem, we could suspect. For confirmation, we can go for ultrasound scan or transrectal ultrasound scan, anything we can take up for the confirmation. Color, I told you, most of the sample have a whitish gray color on ejaculate. And liquefaction, they acquire translucent color. And if there is a yellow tinge or red tinge in the semen, red indicating there is a pinkish color, semen, there is a pinkish red tinge, indicating there is an internal capillary breakage in the testis. Otherwise, if it is a yellowish color, 100% there is a infection. That is indicating the semen color is yellow. Then no such color means it is a normal. And it is like a translucent or like a grayish color. Volume, the volume of the ejaculate should be measured in the centrifuge tubes, 0.1 ml in a graduate centrifuge tube. Low volume may be due to the obstruction of reproductive tract or the congenital absence of seminal vesicle. I told you there is a obstruction, then the volume might be less. It could be the retrograde ejaculation also. Otherwise, the absence of seminal vesicle, then the volume will be less. So volume less means spermatozoa will not survive. Spermatozoa to survive, semen is required. Semen has a liquid as well as the cells, that is a spermatozoa. Viscosity, I told you, this must be determined only after liquefaction. Liquefaction, I told you, what happened? Semen, when it comes out, it will be gel. After 30 minutes, it will become liquid. Once it is in the liquefaction time, then the viscosity of the semen has to be checked. High viscous sample is indicative of infection or the absence of anti-sperm antibody. I told you immunological problem. How to know this viscosity? We should take a glass rod, just put the glass rod into the centrifuge tube, just try to make it. If it comes like a thin, this one, just like a belat paka, then it is a highly viscous. It shouldn't be like that. It shouldn't be sticky. It shouldn't be sticky thing. Then it indicating there's an anti-sperm antibodies are there in the sample. And some bacteriological and immunological. Yes. Yes. Okay. So there must be. Please, if any question, you can interact with me. Closer. Don't interact among yourself. Okay, so bacteriological and immunological investigation is recommended for such patients. Then microscopic examination, the density, it shouldn't be more dense. Amorphous particle means sometimes we can see lot of debris like things. Lot of WBCs will be there in the thing. And amorphous particle indicating there's some infection. Agglutination, so they will agglutinate. Even agglutination is all indicative of some pathological condition. Count, I told you, based on the count only we design and count and motility, morphology. The infertile condition is mainly based on motility, count, morphology. Vitality, how vital they are in the semen, it's also important. So these are all the microscopic examination. Motility, please, you can go to some videos in YouTube or in the internet, how the sperm look like, how they move. Really, you will wonderstruck. And always you appreciate the nature, how well it has designed human, because so mesmerizing. It is a amazing thing. It goes so fast that you can't imagine. So that's a rapid linear movement. It moves fast and with the help of the tail. Sluggish linear or otherwise very slow. Sluggish non-linear, non-progressive means it goes some distance, there it will stop. Non-motile means it won't move forward. It will be moving, it circles. There itself it will be circling. And other than this, the shape of the spermatozoa reflect the normal development of the sperm in the testes, which indicate the normal spermatogenesis process. And here, apart from that, the penetration, addition, fusion of the spermatozoa with oocyte is also very, very important. And in order to know this penetration, addition, fusion, we need to do some function test. Functionality of the spermatozoa will be mainly based on three important tests. That's called as a hypoosmotic swelling test, nuclear condensation test, acrosomal intactness test. These are all the deciding tests to check the functionality of the spermatozoa. Spermatozoa is there, but I don't know whether it is a functional or non-functional. So how to check it? So we need a hypoosmotic. We need to do hypoosmotic swelling test, nuclear decondensation test, and acrosomal intactness test. So this is the hypoosmotic swelling test. So here you can see, I mentioned this is the abnormal. This is the spermatozoa. Human. This is the human sperm. Rat sperm will be different. All sperms are different. Every sperm has a head and tail, but shape of the head will be different in different organisms. So tail will be like a thread-like structure only. This is the human data. So here, this is the head of the human sperm. This is abnormal because this sperm is not responding for the hypoosmotic swelling test. These are all the normal. See, you can see the tail is round here. It is curled. See here, it is curled. All these are the normal. This is normal, this is abnormal. Okay. It is the test is done to check the membrane intactness. You must be knowing the cell has a plasma membrane. If the plasma membrane permeability is lost, then that particular cell is an abnormal cell. The normal sperm will show tail bending, I told you, due to increased hypotonicity, which breaks the disulfide bond in the tail fiber. When we do this test, because of the hypotonicity, then the reagent will enter into the cell. It will break the disulfide bond in the tail fiber, thereby it will become wet. If it is abnormal, it is not responding for the test, then there is no hypotonicity and there is no breakage of the disulfide bond, the tail will be normal. It looks like normal, straight only. It is not normal. Tail is straight. Tail is bent is normal one because it is responding for that particular test. It is just like a positive, negative. How HIV positive, HDLV positive, negative, no? If it curls around, it is a positive, it is normal. If it is not, it is a negative means it is abnormal. If there is any damage in the membrane, the intactness is lost, which decreases the osmotic swelling. Therefore, the tail remains straight without any bending. So next is a nuclear sperm nuclear chromatin decondensation test. Beautiful thing. That's why always I appreciate human body is amazing. What happened, and birth is very special. Always, we never till now, nobody will realize the importance of our birth. And we are not here by we are here by default. We have to, there is some purpose. Because what happened here, what in the genomic DNA, you must be knowing, genomic DNA has the histones. And DNA is wound around the histones. But in case of spermatozoa, DNA is wound around the histone, but it will be replaced by protamines. And once again, before fertilization, it will be once again replaced by histones. In the sperm, the somatic cell histones are replaced by protamine. In the sperm, there is no histone, it is replaced by protamine. And protamines are small positively charged test-specific nuclear protein that facilitate chromatin condensation and compaction. Why protamines are there instead of histone means any mutation happen in a genomic thing, in the somatic cell, fine, the disease or the condition is confined to that particular individual. Something wrong with the germinal cell, it will be carried to next generation. Nature never entertain abnormal creature on the earth because it's a waste of energy, waste of time, waste of everything. Nature never permit such weak and non-productive organisms on the earth. So it has designed in such a way that if it is abnormal, then it is of no use. So always there's a protection for the spermatogonial cells or the germinal cells. That's why any such mutation try to happen, no, because it is highly compact DNA, nothing happens into the germinal cells or in the thing. So it gives a protection from DNA damage. It synchronizes the cell cycle between the sperm and oocyte. It also increases the hydrodynamicity of the sperm. Next is the acrosomal intactness test, very, very important for the fertilization process. The sperm finally, it has to penetrate the zona pellucida. The sperm cap has an acrosome. And acrosome, in the acrosome, there are more than 40 proteolytic enzymes are there. Now they say they have increased the number. They are working on only that acrosome. They got to know that now it has been, when I was doing roughly it is 40, they are telling. But now they are telling recent updated 150 also. They new addition proteins, proteolytic enzymes they have added. These enzymes are involved in capacitation, acrosomal reaction, and chromatin decondensation. And when we do this test, if acrosome is normal, it shows the hollow. If this is C, the acrosome is damaged here. So it is not responding for the test. This is all good. All these sperms are good because they have an acrosome and it binds with a substrate and it shows good response. Acrosome, I told you, play an important role.
The protection you were mentioning in the DNA, is it from it is from protoamines before this slide?
Protamine make a more compactness of the DNA. And this will happen in the sperm because sperm require more protection. Something goes wrong with the sperm, no, it will be carried to next generation. Something goes wrong with the somatic cells, then it is cancer, all these things confined to individual. Such mutation happens in the germinal cell, it will carry to next generation. It will be inherited.
What may be the reason for increase in the enzymes, especially the count you are talking about in the acrosome?
Acrosome, normal individual only having approximately 40. No, not increasing. They are deciphering the remaining proteolytic enzyme. We are thinking that earlier it was you must be knowing there is a acrosine in the acrosome, but not only acrosine. There are so many other at molecular level. If you check, you can see earlier they say it is 40. Now recently they have updated. There are not only 40, even many more are also included. So that's why first we should understand our body only. Then entire universe, we could understand entire universe. So much of information, so much of accuracy to the core we have inside. DNA repair mechanism, then we have tumor suppressor gene, apoptotic pathway, and what are all the things? My God, it is amazing always.
Thank you.
And acrosin is a trypsin-like serine protease that is exclusively found in the acrosome of the mammalian spermatozoa. And acrosin is associated with a zona pellucida binding and zona pellucida penetration. I'll go bit fast because I have not covered all the things. So next comes the oxidative stress parameter. Now all labs are looking for this. Oxidative stress is a condition. It is because there's an imbalance in the antioxidant and reactive oxygen species. ROS you must be knowing. And how the ROS is increased: smoking, exposure to high temperature, pollution, toxic, androgen disruptor, genitourinary infection. Even normal metabolic pathway releases some amount of reactive oxygen species. But what happened? Body is well equipped to nullify, scavenge these reactive oxygen species or reactive nitrogen species by having the enzyme inside. Superoxide dismutase, catalase. There are so many enzymes which will nullify or which will scavenge these reactive oxygen species. This oxidative stress increases by smoking, pollution, cancer, prolonged status of the spermatozoa in the epididymis or in the transit state, immature or abnormal spermatozoa, infection, drugs, all these things increases oxygen species, reactive nitrogen species. What it does, it will damage protein, it will damage biomembrane, it will damage DNA, it will damage lipid peroxidation process. With all this, finally, the sperm is damaged and resulting in infertility. And antioxidants, I told you, superoxide dismutase is synthesized by the body, catalase is synthesized by the body, and there are food-derived antioxidants we can supplement from outside. Always I used to tell everyone, go for this vegetarian diet. In the sense, I don't say don't become completely vegetarian. Occasionally you can have non-vegetarian, but go for vegetables, fruits, green, sprouts, all these things because they have lot of antioxidants. Antioxidant, now if you just Google antioxidant, now you get the big list. Broccoli is an antioxidant. You can see avocado, all berries, blueberry, all berry are very rich in and local, even what is that, guava is rich in antioxidant. Pomegranate is excellent, it is a amruta, it is amruta on the earth. So if you eat pomegranate, all your problem will be resolved. That's a magic fruit, pomegranate.
Amla also, ma'am. Amla also.
There are a lot of locally available things, guava, amla, all these have the very rich in antioxidant. You need not go for apple or you need not go for costly thing. Locally available seasonal fruit. Seasonal, at that particular season, what is available, fantastic. Always go for, even you can take a non-vegetarian diet. I don't disagree with that. You need protein sometimes, but more of vegetarian diet. Now, a lot of research is going on gut microbes. Gut microbes, if your gut microbes is good, your mood will be good. Yes?
Madam, how about the pesticides and insecticides that are being used a lot in these days?
Yes?
Don't you think it is going to affect again?
Definitely 100% it will affect. But what happened? No, always the farmers are the most neglected group. They are dying because of many things. And always I try to link spirituality with science. No, it is a Newton's third law, and we are not supporting them. They are not supporting us. We are giving poison to them. In turn, they are giving poison to us.
This is true, madam. This is true.
And what happened? No, always go for homemade food. Because what happened, many ladies are here, they knew. I never saw any lady cooking vegetable, all these things without cleaning. Everyone, it is like an instinct behavior for females. 3 times they wash, put a salt, all this. No, always they take care of our health, family health. No, But what happen if you eat outside always, all this salad, churmuri, outside, if you eat outside, definitely you compromise with your health. Okay. So always anything is not tasty to your tongue, it will be healthy always. At home, no, it may not be that tasty because they have a lot of concern. They put less oil, less salt, less sugar. But outside, they dump salt, they dump oil. So it will be very tasty for tongue. But health, compromise. So always, all these things, before you cook, you just put it, immerse in a salt water. All the pesticide residues will be removed. And wine and this grape juice is really fantastic, has very, very rich in antioxidant, very rich, because the grape seed has a resveratrol. Resveratrol is a very, very potent antioxidant. That's why many people will promote drink wine, be healthy, be young, all those things. It's fine, not that extent. Why they are telling? Wine doesn't have anything, but wine seed outer layer has a resveratrol. That's the thing. When they prepare wine, it will be rubbed and the resveratrol will be released into the wine. So that's why we have to. And another very important thing for non-vegetarian people, if they eat liver of chicken, liver of goat, liver of whatever, etc., please stop eating only liver. Because liver is the organ, it is a detoxification. All the pesticide, insecticide, all the hormones, antibiotics in the poultry or in the thing, finally will be stored in the liver. Okay. Even if you want to eat liver, immerse liver in a salt for 3 hours, 2 hours. Change water, make it to 2-3 times, then only eat. Otherwise, they do lot of curry, lot of fry in liver, all these things, you're directly taking all the antibiotics, hormones, steroids, pesticide, directly you are taking. And muscle won't store these. Muscle won't store, liver will store. So be careful. Always anything before we eat, we should think what we are eating. So otherwise, our health will gone. So non-enzymatic, lot of things. So I'll skip all this. And biochemical analysis also we have to do. That is semen, we need to check the fructose level in the semen to check the status of the seminal vesicle. And we need to check fructose also indicate a congenital absence of seminal vesicle. Something, fructose level is low, then it is a congenital absence of seminal vesicle. Similarly, citric acid and aspartase, they are the indicator of prostate dysfunction. All these are indicating. So everything, whatever happened inside will be represented outside. And endocrinological, I told you, important hormone, testosterone, LH, FSH. See the level. Variation in these things, it might be due to primary testicular disorder, hypogonadotropic hypogonadism, seminiferous tubule failure, androgen resistance syndrome, then disorder of sperm transport. All these are variation. So this chart is very important. Okay.
Madam, what is this N, all the three
Normal,
N,
normal.
but also normal only, but
Normal.
disorders, okay.
Even then what happened, it is a disorder of the sperm transport, not the production. All hormones are normal means sperm production is normal. It is a disorder of the sperm transport, maybe seminal vesicle and the pathway, it may be spermatogenic ductal or epididymal obstruction. That's all are normal. But here, see, here one is normal, rest of them increased, 2 are normal, 1 increased, indicating particular condition. Chromosomal, there is a numerical variation, structural variation, Y chromosomal abnormality. All these are there. These are all the things. Klinefelters. I have seen lot of Klinefelter in my study. I have seen all kind of variation in both male and female. Ambiguous genitalia. The person has both the testis and vagina. Ambiguous. You can't decide whether he is a male or female. All kind of things, in human we can. So this is a Klinefelters. Then mosaic pattern. Then there is a transformation.
What is the ratio, how many, per
This is the
100 or
percentage.
1000?
No, this is in the percentage. See, I am given in the percentage. Almost 1%, like. Okay. So inversion, deletion, addition, all this. Elongated Y chromosome or inverted Y chromosome, small Y chromosome. These are all the Y chromosomes, variation in the Y chromosome. These are all the karyotypes. What I have shown previous karyotype. See here, there is a problem here in this. There is a problem here. Okay. There's a problem here. This is the fish fluorescent in situ hybridization. This red is a X-bearing sperm. Y is a green is a Y-bearing sperm. Sometimes it is a XXY. Sometimes in spermatozoa it is a XY. Instead of X or Y, both will be there. Sometimes like that. So that signal tells us that abnormality. And Y chromosomal microdeletion, I told you, there are very important testis-specific gene responsible for the normal quantitative and qualitative spermatogenesis, like a DAS, CDY, RBMY, all these things, USP9Y, these are all the things. So this is the Y chromosome and mutation in this AZFA region, B region, C region will compromised with the sperm production. This is a gel profile showing a PCR amplification of the marker in the DAS gene. This is also marker in the DAS gene. In this patient, this is absent. Completely that gene is absent. So then this gel profiling of RBMY1 gene, gel profile of USP91 gene. So we can see in the AZF-C region, 58.3% of the microdeletion, that's the most vulnerable region in the Y chromosome. And ultrasonography also we did to check the testicular volume, assessment of the testicular volume in relation to spermiogram. Spermiogram, we have all the data. So we want to check how it looks like through ultrasonography. And testicular volume was significantly lower, not only in the infertile, but also in the aged one. As person age, the size of the testis will also shrink or degenerate. So this is the longitudinal transfer section of the normal testis. This is the hypoplastic testis. The size of the testis is reduced. And this is a calcification, I told you, because of the infection, calcification in the testis cells are started degenerating because of the calcification. So we shouldn't neglect the urinary tract infection. So hydrocele is the accumulation of the fluid between the parietal and visceral layer of the tunica vaginalis, and there is an imbalance between the fluid production and absorption through tunica mesothelial cell in responsible for hydrocele, means a fluid filled cavity. This is the hydrocele of the testis. And this is the hydrocele, the longitudinal and transverse section, right testis. Similarly, varicocele also I told you, abnormal dilation and elongation of internal spermatic vein and pampiniform plexus of the spermatic cord, and quite common in infertile men. But for the primary infertility, and they can go for a surgery, but sometimes it will relapse. Again, it will develop. So that's why don't put more pressure on the legs. Don't put, weight management is important once again for the healthy thing. Obesity also causes infertility. Adipokine tissues, that will reduce the, hamper the testosterone.
Madam, you mentioned that teachers, male teachers may develop that varicocele.
No, chances.
Chances, why? Why?
Huh?
What may be the reason for developing?
No, more pressure, the entire body pressure will be there for the testis, and it will be, I told you, even our clothing. And not only teachers, long-standing, even people, leg also, they develop the varicocele. Sometimes it will be on the, and people who are working away with, like a drivers and exposed to more temperature, then extra blood supply will be there. Chances are there. I'm not telling they will develop. That is one of the reason. And associated with occupation, all our health issues is associated with our occupation also. Scanning of the normal epididymis. Then we have to check the epididymis size, atrophy of the epididymis. There is a regression of the epididymis. Cyst, there's a huge cyst in the epididymis. And then we can go for scanning, we can use testis and epididymis. But for the seminal vesicle, prostate, vas deferens, transrectal ultrasound scan, that's a color Doppler earlier. This is the transrectal ultrasound scan. They insert the catheter. A probe is inserted and they check. And this is through the anus. Through anus, they will rectum through, they insert this and see like this. And then they will check the status. Normal prostate scanning, then they can see the hypoplasia. And we have reported in first time in India that there is a infertile males have the hypoplastic, that this work has not been done by anyone. This is the hypoplastic. The size of the prostate was small. Then the cyst, you can see the cyst here, huge cyst in the thing. Like if females, they have a uterine cyst, PCOD, polycystic ovarian disease. Like that, men can also have a cyst in the prostate, cyst in the seminal vesicle, everywhere. So the cyst in the prostate. Then calcification of the prostate. Means all this whitish color, no infection. This is infected prostate. Prostatitis means complete prostate gone. Liver cirrhosis like. Complete prostate gone case in this condition. So, similarly, seminal vesicle, there is a cyst. All these are the cysts in the seminal vesicle. Hypoplasia of seminal vesicle. Regression, hyperplasia, enlargement of the seminal vesicle. So finally, ultrasound scan is also very important. We shouldn't neglect. It's a non-invasive methodology and where it gives first order of investigation and provide diagnosis without any side effect. So with all this all-round corner holistic approach, then we can make out whether the person is infertile or fertile or what could be the reason, all this information. So with this, I just wind up. I tried my best to give the outline of the infertility and how all the different parameters are having impact on the infertility. And any questions, please.